Welcome listeners to today's knowledge stream on the thermoscientific EXTREVA ASE and its role in PFAS analysis. I'm your host, Germán Gómez, and I have two experts with me today. One of them is Gopal Bera. He's a staff scientist in the ion chromatography and sample preparation here at Sunnyvale, California, and he has over 10 years of experience in environmental chemistry, PFAS regulations, as well as exposure assessment. He specializes in method development, and he has a strong publication record and strong collaborations with the Environmental Protection Agency at the United States, as well as with multiple academic institutions worldwide. Our second guest is Ed George. He has a wealth of experience in environmental laboratory work. He's our current Environmental Food and Safety Global Key Opinion Leader and Regulatory Affairs Manager at Thermo Fisher Scientific, where he plays a crucial role in shaping regulatory strategies and providing expert insights on environmental and food safety issues. Before we dive in, let's take a moment to talk about why sample preparation is such a critical part of the analytical workflow, especially in gas chromatography and liquid chromatography mass spectrometry applications. So the first thing is kind of like related to ensuring our data quality, right? We all know as analytical chemists that poor preparation leads to poor results. It directly impacts your recovery, it impacts your reproducibility, as well as the detection limits. And in almost always, I would say, it's often the most time-consuming and painful, as well as it's where we made the most mistakes, right? When we do these processes automatically in the workflow. So reducing manual labor definitely help us reducing contamination risk, as well as it help us to even shorten the turnaround time. So in today's knowledge stream, we're gonna cover how the Thermo Fisher Scientific EXTREVA ASE system help us solving a lot of these problems and how it automates the sample preparation workflows for your needs. Let's get started. We're going to answer what is EXTREVA ASE. So Gopal, could you please tell us or explain us what is EXTREVA ASE and what are the benefits EXTREVA could offer to laboratories?
Sure. So as you know, the ASE technology, accelerated solvent extraction, is already superior technology compared to other extraction method because it uses pressure and temperature. While pressure allows your solvent to penetrate deep into your matrices, the temperature helps to transfer the analyte from the matrices to the solvent. Now with EXTREVA ASE, we are bringing that technology up several levels. As you can tell from the name of EXTREVA, EXT is for the extraction and EVA is for evaporation. So we are combining these two steps of sample preparation into one. So EXTREVA can do automated extraction and automated evaporation. Mm-hmm. And then on top of that, you can load 16 samples at one time. Mm-hmm. And EXTREVA can do extraction 4 samples simultaneously. So by doing that, what you're doing is basically you're reducing so many manual steps. And as you know, the more manual steps you have in your method, it's prone to error and contamination. With EXTREVA ASE, you can reduce that dramatically.
And what about for those cases where users typically need to evaporate the solvent? Is that something EXTREVA also solves?
Yes, of course. I mean, EXTREVA is designed to basically do both extraction and evaporation automatically. So you load your sample and you just go away. It will do the extraction, it will do the evaporation automatically, and your sample vial is ready for analysis.
So bring it to the level we need for the instrumentation?
Of course, yeah. So EXTREVA is equipped with state-of-art level sensing.
Mm-hmm. Nice.
Yeah. So it can very accurately see the level of your final volume. It can go up to 0.5, 0.3 ml, or 1 ml or 2 ml.
And what about the versatility? I mean, we can see the instrument has the feature of multiple solvents. Like, is this something users can play with?
So that's very interesting question. Yes, of course. So, you know, PFAS is very interesting and very critical analysis, right? But this instrument is designed to do PFAS and also can do other analyte classes like POPs, because we have 6 channels, solvent channels, and 2 of them are dedicated for PFAS, and the other 4 you can use them for other analysis. So yeah, definitely EXTREVA is very, very versatile.
So you mentioned that EXTREVA can handle 4 samples at a time, and the user can load up to 16 samples on the trays.
Yeah.
So what is like a typical throughput in an 8-hour shift?
If you look at the turnaround time, it's basically can do 96 samples in 8-hour shift.
Oh, that's pretty good.
Yeah, it is.
Thank you, Gopal, for sharing the insights about the key features of the instrument. Okay, so now we're going to learn how EXTREVA ASE is innovative. So Gopal, can you walk us through what are the very innovative features you talked about before in the previous segment about the ACE technology, which we all know is maybe over 30-year-old now. So how is EXTREVA different and innovative compared to the traditional ACE technology?
That's a good question. So if you look at the ACE technology, and if you look at our EXTREVA ASE, the EXTREVA ASE is built on ACE technology, but we improve the ACE technology with a patented technology. So the patented technology is basically gas-assisted and dynamic extraction. What that means is that you are mixing nitrogen gas with the solvent and under high pressure. So what it does is basically break your solvent down into very tiny droplets. And what it does is basically force the solvent very deep into your matrices. And that helps tremendously to bring your analyte out from that complicated matrices. And in terms of the dynamic approach, that's really also a game-changing technology, because it helps with your diffusion, basically. So once you have your certain amount of solvent, you are transferring your analyte from your matrices to the solvent, but then you let it go to your collection valve and putting the new solvent in there. So that helps the transfer of analyte tremendously.
So you're getting like fresh solvent every time.
Yeah. So within that, let's say you have a 5-minute extraction, but within that 5-minute extraction, you are putting fresh solvent continuously and letting it go. So that's dynamic approach is very, very helpful in extraction process.
And let me stop you there for a second. If I recall, Ed, you definitely know better than us, but it's my recall that typically ACE methods were somewhere around 15 minutes or so for extraction. You said 5 minutes extraction.
Yes, it is.
And is it that because of the dynamic?
Yes, dynamic and gas assisted. I mean, like I said, ACE technology is already a superior technology, and when you build on top of that and add these features, it's really untouchable.
That sounds pretty cool, eh?
Yeah, awesome.
I have a follow-up question, Gopal. So that pertains to the level sensing system. Is this something you would normally find in other instruments from our competitors, or is this a unique feature from the EXTREVA ASE?
It's definitely a unique feature of EXTREVA ASE. And generally, people either have to manually look at their levels. There is no way they can do automatically. But with EXTREVA ASE, it's state-of-art technology, basically. Like I said, you load your samples, and you set your levels you want to go. That be 0.5 ml or 1 ml, and our level sensing technology will do that automatically.
Perfect. Thank you.
And very precisely.
So it is my understanding that typically with manual methods, right, like 1633 as written and any other US EPA methods, the materials, labware, glassware, plasticware used for the analysis is typically disposable, right? So worriness about carryover is mostly about the instrument used for detection, not so much about the instrument or the components used for extraction. But in the case of EXTREVA ASE, it's my understanding that some of these components are reusable. Is that correct?
Yes, it is. But we did a very extensive test on EXTREVA ASE to figure out its background, PFAS contamination. So we upgraded our EXTREVA ASE, the previous version, and we upgraded this EXTREVA with big tubings. and stainless steel tubing. So the peak tubing is coming from the solvent bottle to the mixing valve, and from mixing valve to the pump is stainless steel. And when we did that upgrade, after that we tested the EXTREVA PFAS background, and you can see in our application note that the PFAS background is way below the MDL of EPA suggested.
Right, so that guarantees low background from solvents
Yes.
to the fluidic components of the instrument. But
Yeah.
what about like when you actually run samples? Like, have you tested for, we don't have any carryover, and then we can trust the data sample after sample after sample?
Yeah, that's a good question. So yes, of course, we did a carryover test, several actually carryover tests. We ran high concentration samples, SRM samples, and we can see that the carryover is really minimum. I mean, it's way below the MDL. Only thing we can see is like 6 to FTS, but that's also way below the EPS MDL.
Okay,
Yes.
perfect.
Yeah.
Thank you very much, Paul. Thank you, Ed,
Thank
for your
you.
input. So in the previous segment, we heard about how with the new EXTREVA ASE, we could drastically reduce cycle times, right, by virtue of the dynamic and the gas assistance. So I wonder, Ed, how EXTREVA ASE can help labs meet the most pressing challenges?
Yeah, that's a really good question. It depends on the laboratory type. There are several types of laboratories that have different needs, and one of them is the contract testing laboratories, which are generally large multi-laboratory organizations, and what they need is high throughput, and they look at cost per sample very carefully. So they want a very easy return on investment for an automated system like this. That's very important.
Right.
Okay, other types of laboratories like municipalities and state laboratories, they often are looking to comply with regulations and validate their data. Why? Because they are targeting communities, public water supplies that are worried about public health. And that's very important that they get the right result. Okay, other types of laboratories could be, for example, in academic institutions or government labs. And academic institutions really need to publish data, right? That's what's driving them. And so they need to develop methods, publish data, so that way they can get faster to publish with an automated system. And that's really important for them. And the government laboratories are looking at cutting edge research, sort of some unknown contamination, newer organic contaminants. And so the EXTREVA ASE can help them extract them from different types of matrices.
Right.
So that's another type of area that they're interested in. And then finally, food laboratories are another one, food control labs. So EXTREVA ASE can go beyond environmental samples, and that's really important to note here.
Of course.
And so in food samples, where PFAS, it turns out, could be even more of a major concern in food than it is for example, in drinking water. And so these laboratories need to make sure, if you're a food control lab, say for a large food manufacturer, you want to make sure that you protect your brand. Your brand image is everything. Once your brand image is tainted, that's a really problem. And a lot of these big brand names are realizing that PFAS could be a problem, and they're looking at food packaging material and other things that could be causing them a problem.
All right, so if I were to recap here, contract tested labs, EXTREVA offers, thanks to dynamic and gas assistance, a significant reduction, highest throughput. From a standpoint of laboratories, at governments, and food testing, it's reliability, integrity of the data, traceability. And now from an academician perspective, thanks to the automation, fast turnaround in publications because they can process more samples faster. But I wonder, Gopal, how easy is to tune a method in the EXTREVA ASE for these type of scenarios when you're dealing with the unknowns, right? Like we talk about a lot about the regulatory market, but I'm curious, how does it work in terms of non-regulated and unknown new methods, new compounds?
You can play with the method very easily. For example, you can play with your extraction time, first of all, right? So if you look at, let's say, POPs, right? So if you look at some other methods of the POPs extraction, they are generally 15 to 30 minutes. We optimize our EXTREVA to 5 minutes extraction time. And if you look at the recoveries of the POPs, they're excellent recoveries. And then you can also play with your, to move it faster, the turnaround time, you can play with the evaporation. So you can bring down your vacuum pressure. That will help you to evaporate faster. You can play with the temperature and pressure also.
Right.
So it's very easy to optimize EXTREVA.
So if I understand correctly, you can play with different parameters at the extraction and the evaporation. I guess in the extraction that will also include like temperature, the extraction solvents,
and
so on
flow
so
rate
forth.
also.
And the
solvent
flow
flow rate, yes.
And I guess we haven't talked about this yet, but one key component in the EXTREVA, especially for laboratories seeking for either regulated or non-regulated, is the versatility, I guess, of the sample size. Is it something you can talk to us about it?
Yeah, that's a very good question. So the regulatory agencies, they like to have the sample size a little bit high, just to reduce the RSTs. But if you want your sample size to be low, that will help you reduce your turnaround times very significantly. Because once you have your sample size small, then the total volume of your extract gonna go down very significantly, and then you're not gonna need much time to evaporate that smaller volume of extract.
Right, and if I understand correctly, you mentioned about like it was most like a recommendation usually from regulatory agencies, right, to certain sample sizes.
Correct.
But EXTREVA, if I understood, please correct me if I'm wrong, it offers both, right? So the ability to be compliant,
yes,
and also play with the instrument methods and features.
Yeah, correct, because you could, in some of the methods, they specify the amount of sample they want you to weigh into the sample,
right,
for the sample weight. So say 1 or 2 grams of sample.
Right.
But of course, if you're doing non-compliant work, you can use any sample size. And the good news is we have sample cell sizes that are different, from smaller to larger sizes, to accommodate a wide range of sample weights.
So what is the minimum sample size we could pack and run in an EXTREVA ASE system?
Yeah, I'd say around 0.5 grams or even less.
Oh wow,
Yeah,
that's amazing. And what would be the largest sample size could be typically tolerated?
we have 100 ml cells,
right,
so you can fit 20 grams of samples easily there.
Oh wow, that's a pretty broad range.
Yep.
Awesome, thank you very much for this segment. So we have been talking about great features of EXTREVA ASE, all the components and how it works, but let's dig now into PFAS analysis and the US EPA Method 1633A. I have a question for you, Ed. What is the US EPA Method 1633A and why is so important?
Okay, it's a very important method. This method was developed by the EPA Office of Water in conjunction with the Department of Defense, right?
Okay.
And so what they wanted to do was they wanted to address other matrices besides water samples.
Right.
So in the current regulations, the EPA is beginning to regulate municipal water supplies, drinking water, using methods called EPA Method 533 and Method 537. And they are targeted only for very clean drinking water samples that come from these processing systems. So what the Department of Defense decided to do was they needed to look at how do we clean up sites that are contaminated with PFAS. So as you can imagine, PFAS is basically contaminating everywhere. And one of the biggest problems is these wastewater treatment plants have generated a lot of these sludges and other types of materials. It's been applied to land and it's been getting all over the place, right? And so, and we didn't know about it, right, over the years. And as a result, we need to be able to test other matrices. And so EPA 1633 was designed to look at matrices such as soils, biosolids, also non-drinking water samples and tissue samples.
Right.
Because PFAS is pretty much getting everywhere. And so the method is really designed to look at a target list of PFAS, around 40 compounds, by LC-MS-MS. It's a technology to detect the PFAS at a very low concentration. And this is very important because when these sites are cleaned up, they're going to have a standard method to use. And so this is a regulatory compliance method. And so this is a very important milestone to have a common method that can be used across these sites so that the data is comparable. And that's very important, right? And so that's why a compliance method is very important for situations where data is going to be compared site to site, because if you use your own method, right, the EPA is afraid that they could give different results depending on how the extraction took place.
We're standardizing across the entire nation.
Correct, and that was really important for EXTREVA ASE to work with the EPA, with the system. I should say that it was important for the EPA to work with this automated extraction system, the EXTREVA ASE, to use it to make sure it is very similar, or compares directly to, I should say, I'm saying it wrong, but it should compare directly to the method as written. So the method as written is a manual process, right? It's a manual extraction and cleanup, which is very laborious. This process uses the dynamic gas-assisted extraction, which is much more efficient, as we mentioned earlier, but it also is giving the same results as a manual extraction. And that's what we had to show working with the EPA as we developed the method. So
for the audience, you mentioned two methods, the 533 and the 537. How does 1633A compares to those two methods in terms of the target analytes compare?
Yeah, the target analytes in method 1633 are larger. There's about 40 compounds, and there are about half the number of analytes that are in the other two methods. And the other two methods, again, are aimed at drinking water, and they involve a solid phase extraction as well.
Right.
And for the most part, it's a pretty manual process. Right? So, whereas 1633 really starts to break out and look at other matrices. As you can imagine, even non-drinking water samples have a lot of particulates in them, there's sediments in them, they're sort of biphasic samples. And so 1633 addresses that problem by having you separate those phases, where you extract the water sample and you extract the soil sample. So the EXTREVA ASE is designed for the solid samples. That's really what we want to say here. It's designed for the solid samples and makes that process easier. Otherwise, you have to do two steps manually, which is very difficult and takes a lot of labor.
So if I understand correctly, the samples with EXTREVA ASE for 1633A method do not need to be dry. So we could deal with even semi-solids, or sort of like, I understand it's not intended for water samples, but it could be used for kind of like smushy, like wet samples?
Yeah, it can be, and I believe the method actually has you calculate the percent moisture in the samples. And so the method's really clear on how you handle those kinds of samples. And of course, if you have a biphasic sample, so say you have a sample with a bunch of water on top and a big bucket of sediment at the bottom, that's two samples. So you extract the water sample, and then you'll extract the sediment sample with the EXTREVA ASE.
Correct. Perfect. So kind of like to summarize, two of the key difference of this method would be the analysis of a variety of samples that were not covered by previous methods, and an amplification of the target analyte scope.
Correct.
Perfect.
Absolutely.
Awesome. Thank you, Ed, for your insights. All right, so we have been talking about the instrument, you know, what the Thermo Fisher Scientific EXTREVA ASE could offer for the analysis of, you know, organic pollutants in general. We have talked about EPA 1633A method that Ed just explained to us is a very manual method. So, Gopal, key question for you. How does EXTREVA ASE system aligns with the EPA 1633 method? Because, yeah, we heard, right, it's manual, this is automated, like, are we compliant?
I mean, in PFAS testing world, EPA 1633 is a huge step in terms of, it's a very standardized method, like Ed said, right? So, and you need standardized method to compare data to data. Without standardized method, you cannot do that. So in that regard, EPA 1633 is a huge step. At Thermo Fisher Scientific, we spent almost a year to make sure our EXTREVA ASE is aligned with EPA 1633A. Having said that, if you look at 1633A as it is, it has a lot of manual steps. You do the extraction manually, you do your evaporation manually, you do your cleanup manually. With EXTREVA ASE, all we are trying to do is keeping the chemistry same of EPA 1633A. We are bringing automation, and we are streamlining a lot of those manual steps.
Let me stop you here for a second. When you talk about the chemistry, are we talking about the chemistry of the extraction solvents?
Yes, so we are keeping the extraction solvent same. We are keeping all the solvent required to during your SPE steps. We are keeping everything same. We are using the cartridges is recommended.
Yeah.
So we are keeping the chemistry same. We are just bringing the automation, and we are just streamlining the whole EPA method basically.
Wow. And with EXTREVA ASE, do you need to go through the process of evaporation, or is this something that?
Yeah, that's a good question. So if you look at EPA 1633A, you will see that after the extraction, you basically dilute it and then evaporate down, and then you dilute again. So with EXTREVA ASE, what we are doing is, after the extraction, we are just diluting it to match the organic content below 20%.
Yes.
And then you can go directly to SPE phase.
Right. And
so by doing that,
yeah,
what you are doing is basically eliminating the evaporation totally.
Right.
And that way you can reduce your turnaround time significantly.
Which is my understanding exactly. For many workflows, regardless of the target analyte, evaporation usually it's very prone to error, number one.
Yeah.
Number two, not that EXTREVA cannot do it, of course.
Right.
We typically use it for other type of compounds, but in this particular scenario, it's you demonstrate it's not needed, right?
You don't need it, yeah,
Correct.
basically. And you, like you said, by doing that, you're reducing the sample handling.
Yeah.
And when you reduce the sample handling, you are basically reducing the risk of contamination also.
So you talk about the evaporation, right? So how this, tip drastically reduces time. But so how does the 1633 method perform on the EXTREVA ASE compares to the manual method in terms of labor?
Oh, that's a good question. If you look at the 1633 as it is, it takes about 12 hours of manual labor.
How many?
For, I would say, 16 samples.
12 hours.
Yeah, because especially for, I'm talking about just soil, sediment, and biosolid, not even talking about fish tissue.
Right.
Because for fish tissue, the
or
extraction time.
Animal tissue in general.
Sorry?
Animal tissues.
Yeah, animal
Yes.
tissue in general. That takes long time.
Right.
With EXTREVA ASE, you can bring your manual labor almost 70%. Because
70%.
70%, because it's all automated. You're doing the extraction automatically. There is no evaporation.
Right.
And then you can do your cleanup through SPE semi-automatically.
So we are expecting at least 70% reduction
In
manual labor.
Yeah.
And what about the turnaround time?
In terms of turnaround time, I'll say it's about 30% less.
So with that in mind, we're talking about like something that is very important, I guess, for contract labs, for instance, right? Yeah. They care a lot about return of investment and how much money they can, make when analyzing a given sample. But what about, you mentioned this before, right? Like in terms of some others, manufacturers of instruments make claims about compliance. But how is it to get a compliance for the instrument? Like you mentioned a year working on doing a thorough testing.
Yeah. So
can you walk us through that one?
Yeah, of course. For us, it's very important that we validate our EXTREVA ASE in a very robust way.
Right.
So to start with, what we did is internally to look at our instrument to make sure the sensitivity wise is good, our LCM SMS. From there, we basically look at our EXTREVA ASE to make sure there is no PFAS contamination, background contamination. And from there, we started collaborating with EPA.
Right.
Actually, we developed our study plan.
Right.
Along the way, they advised us to develop our study plan.
Right.
And once we have a solid study plan, we basically started doing analyzing the samples. The samples sent by EPA or EPA collaborators.
Correct.
We had soil samples, we had biosolid samples, we had tissue samples.
Right.
On top of that, we also have SRMs.
Which are certified?
Certified reference material from NIST, mainly from NIST.
Okay.
So what we did was we compare side by side.
Correct.
So we ran those samples through EPA 1633A.
As prescribed.
As prescribed.
Right.
and then we analyze through our EXTREVA ASE
With the instrument,
based method,
Right.
automated basically version of it.
Correct.
And we look at side-by-side data, we compare, we saw excellent similarities in data.
Perfect.
Basically, there is no significant difference in those two data sets.
Right, and we're going to dig into a few more details on, the analytical values and validation process. Thank you very much, Gopal. So we were talking about like the importance of how EXTREVA ASE, in terms of performance when compared to EPA 1633 and the collaborations that have been ongoing with the Environmental Protection Agency of the United States. But I'm curious at how does EXTREVA and what are exactly customers looking for when thinking about an automated solution for PFAS?
Yeah, I think the primary thing they're looking for as a laboratory, right? They're going to look at obviously the cost of the instrument compared to the labor it's going to save them, right?
Right.
So in automation, that's what you're always trying to weigh. So it's really important that, again, a return on investment proposition for the laboratory. And so what they need to decide is whether or not having hands-on labor is going to be, supplanted with an automated approach. But at the same time, they need to make sure that the data that they get when they do supplant that is very accurate and it's efficient, right, in terms of the operation. So the analysts would need to be trained on the automation approach, and then at the end of the day, generate data that's more reliable and more accurate and precise, right? So in precision is a big problem in sample preparation, right? So if I have 2 or 3 or 4 different operators, when they come up and do a manual process, they're going to do it a little bit differently, and as a result, the precision will be worse. And that's a real problem for laboratories that are trying to generate the same data for their customers. So the automation really gives them that ideal situation of getting that precision under control. It takes the human error out of it because you're going to have that with different people and training them. So in these EPA methods, you have to do what's called a demonstration of capability. And that basically tests the operator's ability to do the method manually and get results that are proper in terms of accuracy and precision.
Right.
The automation will help the operator ultimately attain this demonstration of capability a lot faster because it's an automated process. They don't have to worry about transferring things from one place to another, losing sample, contaminating the sample, getting more background in, because if that happens, they're going to fail their test and they won't be able to run routine samples for compliance monitoring.
So if I were to summarize this, would you agree that it's a great tool because even for untrained labor, I mean, there is very little these new employees or someone getting into the method will need to learn, since the instrument already kind of like automates most of the tasks that need to be performed?
Yeah, that's absolutely right. And the other important thing is that the methodology has been figured out for the operator. So there's no guesswork. When the system is put into the laboratory, the method is already set up, and the customer can start using it right away without doing a lot of tweaking or research. And that's a really important point for these routine labs that are trying to get work done, especially with operators, several different operators are trying to train.
Right, and this would be a segue just for you, Gopal. So it's my understanding that the method that was currently developed in our lab for PFAS testing, it's transferable across the matrices that are currently of concern as per US EPA 1633A. So talking about method, trained labor and method transferability. So just to start, I wonder, Gopal, is it the method that was developed on EXTREVA ASE for PFAS analysis transferable across the different matrices that are covered by US EPA 1633A? Like, can we use the same method for, let's say, biosolids and tissue?
So we have 2 different methods. One is for solid, soil, sediment, and also biosolid.
Okay.
That's the one method. And for tissue, it has a different method compared to the solid. So if you look at 1633, we are trying to align with 1633, right?
Okay.
So for the solid, you just extract with basic methanol.
Right.
That's all you do.
Okay.
For tissue sample though, not only you have to extract with basic methanol, but you also have to extract with acetonitrile.
Okay.
And that you can do with our EXTREVA too. Yeah.
Okay. Is this a combination of solvents, or does the instrument offers the feature where the user could load independent solvents?
It can actually do independent solvents.
Okay.
So you extract with basic methanol, and in the sequence you can actually design your sequence that it's gonna extract with basic methanol, and then it's gonna go to acetonitrile and extract with acetonitrile automatically.
And then
you don't
both
have to do
extracts are combined in the
in your collection vials. Yes.
Okay. Perfect.
Everything is automated. Once you set up your sequence and method.
So that's great to hear. So in terms of comparability, we're going to have two methods, one that covers solid by solids, one for tissue samples. What about efficiency? Because I know that would be one of the greatest concerns for most users. Like, how did the instrument perform compared to the manual method?
That's a good question. So since we are keeping the chemistry same, so in terms of the extraction efficiency, there should not be any problem, and we didn't see any problem.
Right.
When we were comparing the two methods side by side, we can see that at least to the least, basically, our EXTREVA ASE method is matching the requirements by EPA 1633A. So when we were developing our study plan, we had a guideline that when we will compare the data side by side, the average recovery has to be within 30% of EPA 1633. That just as a guideline. But when we are comparing the data, we can see that our recoveries are same or better.
And this is across all the
matrices. Yeah.
So that's great to hear that the instrument has a great performance across the different matrices. But I'm curious, because when someone is transferring from a manual method that has been thoroughly tested in multi-laboratories across the country, how the EXTREVA ASE does in terms of reproducibility?
Yeah, so in terms of reproducibility, I mean the Parson RSD, if you look at the two data side by side, you will see that EXTREVA ASE does a really good job in terms of RSD percentage. So for most of cases, soil, sediment, and biosolid and tissue, you can see the RSDs are at least equal or better than EPA 1633.
Well, that's great to hear. And is there anywhere where we could find, like, our viewers could get an access to this, to preliminary of this data?
Yes, we have published application note, and there are a couple of manuscripts coming in our way too.
Right, and I think one of them was published just less than a month ago,
Waters,
right?
Yes.
Yes, so the American Chemical Society, EST and Water. Okay, perfect. Thank you very much, Gopal. I invite you, all the viewers, to take a look at the manuscript on PFAS analysis with EXTREVA ASE. So we have been talking about all the features of the instrument and meeting performance of US EPA 1633A. But one thing that worries me, and I guess worries many users, is about the installation of the instrument, right? And whether the instrument is truly PFAS free, right? So we see that claim all the time among our competition. And I'm curious at what care is needed for the installation and what care is needed to deliver a PFAS-free instrument.
Yeah, a real critical part of that is going to be the consumables and solvents that are used in the laboratory. So backing up a little bit, we recommend solvents and consumables that we've tested and we know that are essentially PFAS-free. In other words, they won't generate as much background when you use these consumables, such that you will surpass, for example, the method requirements in the EPA methods. So it's really important that those blanks are clean, right? So everything contributes to it. So it's important that the instrument's installed in the right place. So there's a pre-installation manual which talks about the best ventilation and the best place to put it in your laboratory, and that's really important, along with the solvents and consumables that we recommend. And Thermo Fisher Scientific does sell all the consumables and the solvents needed for this. Another very important thing is that we also have a white paper that basically outlines the installation for our field service engineers to ensure that they're installed properly and working properly. And the next thing they do is they often rinse the system out, right? So they will rinse the system out to make sure that there's no PFAS contamination coming from the system. So it's like a system check, right? So they'll rinse it out for a couple of hours to make sure that all the components are completely free of PFAS, and then the system is ready to use for the end user.
So basically, after unboxing and field service engineer checks all the components, they rinse the system for a couple hours, and afterwards instrument is ready.
The instrument's ready to go, yes it
with
is.
a background that is compliant with 1633A.
Yeah, that's the most important thing to the end user.
Right,
Yep.
and where our viewers could find this information about consumables that they could be used on EXTREVA ASE.
Yeah, there's websites on Thermo Fisher. We have a consumables website that's dedicated to PFAS materials.
Okay.
And so I don't know what that website address is right now in my head, but indeed we can have that edited in.
And I would guess, Gopal, that could also be found on the
We have
application notes.
Yeah, we just published one application note, and you
Okay.
can find the list of consumables and supplies
In the
there.
application note. That's a
Excellent.
good point.
So it could be either the Thermo
On the
Fisher
website.
Scientific PFAS dedicated website or the EXTREVA ASE application note.
Yes.
Excellent. And talking about that one, Gopal, kind of like a segue question here. Can someone claim an instrument is zero PFAS?
I mean, PFAS, like Ed said, PFAS is everywhere, so you cannot say it's zero. What you can say is that whether it's compliance with your intended method.
Correct.
For this case, it's EPA 1633. So as long as your PFAS backgrounds are below EPA's MDL,
Right.
it should be good to go.
Okay. And
in this case, our EXTREVA is below, has PFAS background below EPA's MDLs. Yes.
so based on your experience, do you have any recommendations for our users, right, in terms of, the whatnots in order to, because, I mean, you and other colleagues have thoroughly tested the
Yeah.
EXTREVA ASE for PFAS analysis in a variety of samples. We're curious what should be not done, or what would be your recommendation in terms of lab space and other consumables, in addition to the ones that Ed just mentioned.
So in our case, what we did was we tried to simulate the user cases. What that means is that we were testing our method in a shared lab space. So when we did that, you will see time to time some of the PFAS background coming from variety of sources because of the high traffic in the lab. So what I will suggest to our customer is to basically, if possible, to install the instrument in an isolated space. That way there is no high traffic basically.
Right.
Because if you do that in a high traffic area, you will see PFBA especially in your blank.
Correct.
Because PFBA is even in the air, right? So
Right.
yeah.
And so in line with Ed's recommendation, would you also suggest essentially for our viewers to follow up the recommendations that are currently in your...
Yes, of course. Otherwise, your background will be low and will be under control, but time to time you will see some of the contaminants in your blank if you don't do like isolated space.
Excellent. So for our viewers, is the expectation in the coming months a white paper on full recommendations on PFAS with EXTREVA ASE will be presented. Since we started the conversation earlier today, something being in my head, it pertains to the no need of evaporation when you utilize the EXTREVA ASE versus the method as prescribed, and not because the EXTREVA ASE cannot accomplish this task. So can you walk us again why evaporation is not needed in this method that was developed by Thermo Fisher Scientific?
So as I said before, that with EXTREVA ASE, our primary goal is to basically bring the automation and basically streamlining the different steps in EPA 1633 while keeping the chemistry same.
Right.
So if you look at the EPA 1633A and their evaporation step, what they do is basically after the extraction, they evaporated it down and then they bring the volume up. So for us, it's like a redundant step.
Okay.
You can do it the other way. So what I mean is, when you have your extract, you can basically dilute it with DI to bring your organic percentage below 20%.
Right.
What that will do is basically that you are bringing a little bit larger volume to your SP step.
Right.
But if you look at the overall process, we are still reducing the turnaround time significantly.
Right. And when we talk about reduction of turnaround time, what is that? If we could quantify it, what would be that number?
Overall, the turnaround time is 30% less with EXTREVA ASE.
By not going through this redundant step of evaporation. What else do you gain by not evaporating?
Yeah, so if you eliminate your evaporation step, basically you are reducing your sample handling.
Right.
And in terms of PFAS, I mean, if you can reduce sample handling, you are basically reducing your risk of contamination.
Right.
So that which is a huge problem for PFAS testing.
And I would guess also some of these compounds tend to be a little bit volatile, right?
Yes.
So,
yeah. So yeah, if you eliminate your evaporation step, you are reducing that also.
Right. So even with an automated system, that's the intrinsic nature of these molecules, I would guess, right? So it's expected for that to happen, right? So not to deviate from the main topic of today's conversation, right? So we know that for PFAS, evaporation is not needed. It would be a redundant step. But Ed, will be those cases where we could use the full potential of the EXTREVA ASE system and where evaporation could actually be a game changer?
Yeah, what's really important are methods like 8270, where you have to do solid samples,
Yep.
or dioxins, okay, because those require very low detection limits in terms of regulation.
Right.
Dioxins are regulated at subpart per trillion levels.
Right.
And so that evaporation step will be necessary to make sure that you're able to meet the detection limits in those methods. So what's great about the EXTREVA ASE, however, it's like walk-away evaporation. It will take care of the evaporation step, and what's nice is when the evaporation gets down to the level it needs to sense and stop, it will stop automatically as it has a really advanced detector to sort of stop that evaporation. And it'll also leave it in the autosampler vial.
Correct.
That way you can take the autosampler vial and walk it up to your LCMS-MS or GCMS-MS system and do the analysis. And so that's really important because, again, avoiding the transfer and that evaporation step is really critical to be able to hit those detection limits in those two particular methods.
That's excellent. So basically what we're hearing is that EXTREVA is a very versatile instrument. I mean, definitely cases like PFAS, we won't go through the process of evaporation. In cases like dioxins, definitely we would like to go evaporation route in order to reach the proper limits of detection. What do you think is the future of this instrument in PFAS world, Ed? Like, Gopal and the team have done an impressive work covering all the matrices related to EPA 1633, which is mostly environmental testing. Where do you see us going with the instrument, knowing the capabilities?
Yeah, what's really nice is that the regulations are starting to branch out, right? So obviously it started with the Environmental Protection Agency when they knew that PFAS was everywhere in the environment.
Right.
That concerned them a lot.
Right.
Now we're starting to look at other matrices like cosmetics and textiles and food contact materials, even some of this dark matter and lithium batteries. So a lot of matrices that would apply to the EXTREVA ASE for PFAS extraction. And there's a big concern because we don't know how much PFAS is in these materials. All we know that it's present, and we don't know how much is getting into landfill leachates and other places where it's adding to the environmental contamination. So not only environmental, but all these other sources. We also have food, which is just beginning to be regulated, and it's looking like when you start to look at food, that could be a more major source of PFAS than even water. So that's sort of coming out now in some of the later academic circles. And so it's a real concern. And so the EXTREVA ASE is well positioned to automate extraction of those types of materials.
That's excellent, right? So we're definitely looking forward to a world where EXTREVA not only solves problems from an environmental perspective, but also from consumer products and whatnot.
Yeah, because these companies are really trying to look at their process, and they're being forced to remove PFAS from their raw materials.
And guarantee that.
And guarantee, but it's not easy. Why? Because PFAS have very interesting chemical properties. They're anti-slip and anti-moisture resistant, right? And so finding a material to replace a PFAS is very difficult. Just like in the past when they had the RoHS initiative where they were looking at lead and cadmium in circuit boards, the same thing's happening here. How do we replace those materials? But then how do we replace it with a safe material, a safe and effective material? That's the critical thing. And the semiconductor industry is really trying to look at that as well. So big companies are saying, how do we get rid of it out of our process? Because what's happening is that PFAS being used to create some wafers and other things in the semiconductor industry is ending up in the wastewater and being dumped back into the environment. So it's a big circle of problems that need to be solved here.
And it's a big circle where EXTREVA ASE could intervene and then provide a solution for our customers. Awesome, thank you very much. So EXTREVA ASE, as we know, it's the evolution of a legacy instrument, which is the AS350, right? So we're curious on how EXTREVA ASE compared to our competition, right? I mean, EXTREVA, in a sense, is either a pressurized liquid extraction or also called by others as pressurized fluidic extraction system. We call it accelerated solvent extraction. In general, it's three terms used for essentially the same concept, right? A pressurized liquid to extract from a matrix. So first of all, Ed, I wonder how EXTREVA ASE differences from ACE 350. What are the key aspects you would say distinguish one instrument from the other?
Yeah, well, the main one, of course, is the fact that the EXTREVA ASE is PFAS ready.
Right.
So
Yes.
the earlier ACE models aren't PFAS free.
Right.
So the components haven't been checked out. And that's a really important aspect on the EXTREVA ASE, that it's been completely checked out so that you can generate a PFAS free blank. That was one critical aspect.
Right.
The other one is the dynamic pressurized gas extraction that was alluded to by Gopal earlier.
Yeah.
And the fact that it reduces the time to extract the sample significantly, as well as reduces the solvent. And that has a big impact. Why? Because therefore you're not throwing more solvent in the environment, so the environmental footprint is smaller for the EXTREVA ASE, right? You're not generating as much solvent because you need less solvent with this system to extract your sample efficiently.
And when comparing to other vendors, like that also commercialize pressurized liquid extraction systems like the EXTREVA ASE, what would be the advantages EXTREVA ASE have to offer?
Yeah, the biggest advantage is the integration of the extraction step and the evaporation step. It's all integrated together. Because once you separate them, as I mentioned earlier, you're going to have chances for contamination and also just increased time and labor dealing with it. And so having it as a almost true walk-away system, that's the important point, right? That you're going to set this up and you can do these samples, very easily and walk away from it without having to intervene is a critical step. Other types of instruments that are on the market, a lot of steps and modules, adds more cost and adds more labor.
Right. And if I recall from previous conversations, we're talking about 16 samples is typically the walk away for EXTREVA ASE with a throughput up to 96 samples.
Correct.
In terms of sample sizes, I know we touched this in the previous segment, but I'm curious in terms of what would be the sample cell sizes? Are we similar? Are we different compared to our competition?
Yeah, the sample cell sizes we have are 1 mil and 10 mils, and I believe up to 100 mil sample size, right? So we can cover a wider range than a lot of the competitor systems out there. They have, some of them are very small sample sizes they can handle.
Correct.
But in general, we handle, I think, the widest range of sample load. So the instrument's very adaptable to what you need to extract. And that's really important, again, because you have EPA samples that require a certain weight because you can't deviate from that prescriptive method. But then you might want to go down to very low concentrations of, say, some textile or some other type of material that you're trying to extract. So having that variability and the ability to change the sample size is really important in the EXTREVA ASE that I think is different than our competition.
Yeah, that's a very good segue actually for you, Gopal. So I'm curious, right? So I asked this question to Ed before, but where do you see the future of EXTREVA ASE for PFAS analysis?
One thing you can do in future is to reduce the sample size.
Right.
That will, especially for I would say for tissue samples, because in tissue samples, especially with high fat percentage, I see a lot of metric interference from lipid, right? So it basically, you will see ion suppression in your analysis. So in that case, you need to basically use smaller sample size. Preferably, yes. As long as you can control your RSD percentage, definitely you can do that with EXTREVA ASE, reduce the sample size.
Excellent. So basically what I could gather is it's a very versatile instrument that it could be tuned depending on the needs, whether it's throughput, right, at the right detection limits, or whether I could also further reduce the sample size in order to increase even more the throughput, or increase the sample size in order to decrease my levels of detection and being able to see even to lower limits.
Correct.
Thank you, Gopal, for your insights. So the key takeaways from today's conversation in this knowledge stream, it shows us how EXTREVA ASE truly simplifies PFAS workflows with automation and a reduction in contamination. It allows for at least 30% reduction in turnaround time and 70% or more in labor reduction when compared to EPA 16338 as prescribed by the Environmental Protection Agency. It is built for compliance and performance, and it's completely aligned chemistry-wise with EPA 16338, with the exception everything else is automated. And it's designed for every laboratory, as we saw today. It can go from the innovators who desire to play with the tunability of the instrument, to the compliant labs, or even to the contract testing labs that are willing to scale and to gain even more throughput. It's definitely a tool that is ready for the future, and it could be implemented as of today for environmental testing, and in the future also in food testing as well, and textiles and many other applications like the semiconductor industry. Our call to action is for you to please visit thermofisherscientific.com and take a look at our application notes, publications, as well as the EXTREVA ASE apps library, and of course to my colleagues Gopal Vera's recent publication in the American Chemical Society EST and Water manuscript.